So our next speaker will be Zachara, and he served as a EMSL Associate Director previously. So earlier on, Steve introduced the pioneer with a different intonation, but I really think John is a true pioneer in subsurface biogeochemistry. He has won E.O. Lawrence Awards, and he's one of the brightest PNNL laboratory stars. It's my great pleasure to introduce Zachara. He will tell us about subsurface science.
Thank you very much. Well, as a pioneer, I'm going to try to dust some dirt out of the hair and do everything else that's necessary for an old pioneer to give an understandable talk here today. So I'm going to be talking about the subsurface environment today. And first of all, I'm listed as the author here, but it's important to recognize that the work that I'll be talking about has been accomplished with many collaborators. That's the essence of the EMSOL facility. Both collaborators from externally, internally, and with the highly qualified EMSOL staff that's here that enables the measurements that I'll be talking about. So I won't be identifying specific people individually unless it slips out, but there must be close to 50 or more people involved in this kind of work, and they're dedicated people with very, very good ideas. So this is the subsurface environment. Probably most of you understand what that is. It's the below ground region. And the below ground region includes soil. Let me get this. Soil, deeper unconsolidated materials, down into fractured bedrock and down to the center of the earth, basically. And so this is an important area, as you well know. It's the source of much potable water. It's an intimate connection with surface waters. They basically function as a common unit. It's a location of much of the Earth's biomass in terms of microbes and invertebrates and other activities, and is also a major region of sequestered organic carbon, which is of obvious importance in terms of climate change. It's also the medium for agriculture, for plant rooting, and it unfortunately has been a common and ubiquitous location for waste disposal, as we well know from the Hanford Site. And in some areas, it's a host for energy deposits, such as gases, oil, and the like. So it's important to our nation's national security, energy sources, but also to human health in terms of biogeochemical cycles that occur in this environment. And it's also important to recognize that the subsurface is really an ecosystem. It's a very complicated ecosystem. And the nature of that ecosystem has really been elucidated by DOE research and by BER's research over the last 25 or so years, beginning before EMSOL was in place and continuing through their ongoing research programs to this day. And so this is an area that DOE has made probably the largest contribution to in terms of other agencies. But as we think about the subsurface as an ecosystem, it's important to recognize that its function is controlled by the coupling of geochemical, microbiologic, and physical transport processes. These do not occur individually. They're highly interactive. The physical environment controls the chemical controls the physical, as do the microorganisms. Another thing to recognize that it's influenced by complex hierarchical physical structures, including soil structure, including joints and fractures in rock, including all sorts of microcracks, macropores, root channels, all of this stuff. And the critical thing about these structures is that they direct and channel the movement of water. And water is what moves things around and really supplies reactants to reactions and takes away the products. Another important thing is that small and large-scale phenomenon are coupled. That the processes occurring at the very smallest scale, such as perhaps in this region, influence the macro scale. But the macro scale influences the micro scale as well, in terms of boundary constraints, environmental conditions, and the like. So EMSL was devised from its inception to look at scales, scale dependency, but also phenomena that go across scales. And another statement of I think, Allison's figure, and one that's critically important to subsurface science, is the concept of developing a facility with multimodal instrumentation and modeling capabilities. A lot of different things that can be applied to a problem, because our problems in the environment are very complicated. There's lots of different chemical agents, different physical structures, different scales, different nuclear and electronic states for spectroscopies. And it's critical that one has a range, a significant range of instrumentation to elucidate these kind of things. And it was recognition of this in the initial planning of EMSL that led to the suite of instrumentation that was selected for the environmental sciences. And 20 years ago, when EMSL opened, EMSL was a unique facility, at least for the environmental sciences, in terms of having the largest and most comprehensive suite of instrumentation to address a state of science issues in environmental science. Now, this is just a simple representation of a few of the capabilities: transmission electron microscopy, cryogenic laser-induced fluorescence spectroscopy, Mossbauer Spectroscopy to look at iron mineralogy, and then a subsurface flow and transport facility where microcells can be fabricated to look at microscale transport processes, and then larger flow facilities to study the impact of microscopic and molecular processes on macroscopic outcomes. And so a critical aspect of EMSL has been this ability to cross scales from the atomic to a scale that's really relevant in the field, taking materials from the field, being able to study them, but also be able to apply understanding and models developed at these smaller scales to this larger scale. And that has been a hallmark of EMSL since the facility opened 20 years ago, and it continues to be. And in the presentation that follows, I'll be trying to emphasize this multimodal concept and how different types of instrumentation have been used in a comprehensive but integrated way to solve a few important problems. Now, when EMSL opened, that concern over legacy contaminants on DOE lands was an important driver of initial subsurface science research. And at that time, BER had a number of programs that were looking at legacy contaminants. The first was the NEIGHBOR program, Natural and Accelerated Bioremediation Research. And this was looking at microbiologic processes regulating polyvalent metal speciation and mobility. There was also what was called the Environmental Management Sciences program that changed into a BER program, the Environmental Remediation Sciences program, that was focused on contaminant and water movement issues at Hanford, the Savannah River site, and other DOE lands. Later, there was another BER program that was called the Integrated Field Research Challenge Sites. This was a program that looked at larger scale field sites to develop a system scale understanding on contaminant dynamics and bioremediation. And this included sites at Rifle, Colorado, Oak Ridge, and Hanford. There's also been DOE BES Geosciences research that actively utilized the EMSL facility looking at environmental molecular processes. And finally, there has been Hanford work that I term wraparound science. I'll be talking about this, that represented a collaboration between Hanford Site scientists and members of the EMSP and NEIGHBOR programs. So when EMSL started, there was a large infusion of users from these programs that saw the incredible capabilities in EMSL and really utilized those and integrated them into their research. And in the subsurface science area, that this led to a real flourish of publications on microscale and molecular processes, really underpinning environmental issues. Now, I'd like to just identify up front many of the accomplishments that work has really done. And then as we go into the presentation, I'll be picking a few of these and really showing some of the details in terms of instrumentation used, the nature of the problem, some of the results, and really the implications to problem resolution. So in terms of subsurface science research, I guess we can say that these two areas represented major areas, thrust areas over the past 20 years, environmental geochemistry and environmental microbiology. That it's really actually hard to identify high-level accomplishments because there have been many. And so it comes down to value decisions, one's own bias and opinions, but I think that these are some that many would agree are quite important. First of all, EMSL has provided techniques for molecular speciation at concentrations for well below those of certain of the light sources. And so close to 2 orders of magnitude or more below the synchrotron facilities. Now, having these detection limits have allowed the development of fundamental relationships between contaminant speciation, another word for that is chemical form, or the nature of the contaminant, where it's associated in terms of mineral species, and details of its physical environment, and linking this information with macroscopic behavior, meaning behavior at a field-relevant scale. And this has led to the development of a whole new class of models that describe the interaction between physical structure and chemical and microbiologic processes. The research has also identified the identity and reaction rates of what we call reactive iron species and phases in complex heterogeneous environmental materials. And it turns out that these phases and species are some of the most important environmental reactants that control the transformation of organic matter, of contaminants, and lots of other things of interest to mankind. In terms of environmental microbiology, the facility made major contributions to understanding the mechanisms of electron transfer between microbes and mineral matter. It also established the reactants and products of microbial processes at the subcellular, cellular, and niche scales, supporting reaction and metabolic modeling and understanding biomineralization pathways. This whole area of research then provided the underpinnings for the use of subsurface microbiologic processes for bioremediation and the passive treatment of contaminants in many different areas. More recently, EMSL capabilities are providing insights on the molecular forms of organic carbon, driving microbial processes of different type and function, and I'll be mentioning those as well. So let me begin early with some of the initial work that was done in EMSL for the NEIGHBOR program. The NEIGHBOR program, again, was looking at the interactions of microorganisms, subsurface microorganisms with mineral matter, with implications to bioremediation and transformation of legacy contaminants. Early on, the facility showed incredible promise in terms of imaging the interactions between a microorganism, shown here, and mineral material that had been generated by its activity in connection with other mineral phases. You can see the organism here, you can see the size of the precipitated materials. Electron diffraction allowed identification of those kind of materials. Transmission electron microscopy, and note the scale here, these are nano-sized precipitates, showed us the domain structures and sizes of these things, leading to hypotheses on how microorganisms would form them. And it's important to recognize that this phase, magnetite, which resulted from the transformation of an oxidized iron phase, is highly reactive to polyvalent contaminants, chromium, uranium, technetium, plutonium, and others. And this kind of research, recognizing the capabilities that EMSL had for this kind of imaging, but other things as well, drove a tremendous amount of research looking at how organisms change mineral materials, how they initiate the formation of new materials, and looking at reactions that occurred on the surfaces of these, perhaps retarding the migration or slowing the migration of contaminants such as these identified below. But early on in EMSL, there were questions on well, how can the facility really be used to address a large, important, unresolved question that had previously been, let's say, unresolvable with techniques that were available at the time? One of those questions that resulted from neighbor work was the question, how do microorganisms dispose of electron density resulting from the metabolism of high energy reduced organic compounds to energetically stable iron compounds in the mineral world? And shown over here on the left is a planar form of a mineral called hematite. It's an iron oxide. You can see that the surface of it is colonized by organisms. They're attached to the surface. They're interacting with it in certain ways. And if we take away those organisms and look at the surface of that, that material with a microscopy that's present in EMSL, scanning probe microscopy, one sees some fairly unique features on the surface of this. Dissolution features that have directionality, that have structure, and if we could see the organisms in place, you'll notice this is the imprint of an organism here, this is probably another one, this is another one, have direct relationship to where those things were sitting. And so for many years, if not centuries, there had been questions, well, how do these kind of organisms do it? And really, what is the molecular process or mechanisms by which these kind of things are accomplished? And so with support from EMSL and the neighbor program, BER, we initiated the first grand challenge that was performed in EMSL. And this grand challenge was focused on what we call the Poram cytochrome model of electron transfer, which at that time had not been documented and was only beginning to be hypothesized. This grand challenge involved about 20 investigators from outside of the lab, international participants chosen for their expertise. And this was what initially was a 3-year program that finally took close to 5 to 7 years to reach scientific completion. It was a very exciting program, and it was one that melded concepts of mineral surface chemistry with metabolic cycling within microorganisms, and with protein structures and function, as shown here on the right. The Poram cytochrome model, as it stands now, is one where organisms release electrons, electron density, from the metabolism of reduced carbon compounds that came really from photosynthesis and the sun. There's stored energy in those compounds. They extract energy from the conversion of those compounds, releasing electron density. And the big question was here is, how does electron density get from the internal part of the cell, where that metabolic process is going, out through the outer membrane and to the mineral material? So as it turns out, that a protein complex involving 3 distinct proteins, mTRA, which is a multi-heme cytochrome, it's a metalloprotein, combined with what's called a Poram protein, further linked with another metalloprotein on the surface, is responsible for this. And this model has been shown, once verified through the EMSL Grand Challenge, to be a broadly applicable model of extracellular electron transfer, valid not only to the organism types that were studied, but also to other organism species, and also to iron oxidation, where electron transfer actually flows reverse. This is the reductive flow, but it can also flow this way. Electrons can flow the opposite way, also generating energy for the microorganisms, but using this same protein complex or molecular wire. Now, time will not allow me to show you all the different techniques that were utilized here to study this. But issues of thermodynamics, protein structure determined by crystallization and X-ray crystallography, protein mineral surface interactions explored by molecular modeling with the supercomputing facility here, electron paramagnetic spectroscopy to look at the angles of what we call heme groups with the surface, that's critical, issues of solvation, kinetics, and a lot of other things. This was a very comprehensive activity. Now, in addition to all the science, This Poram cytochrome model was confirmed through a number of activities that I'll show here, just very briefly. You can see these yellow and black dots on the surface of these features. This is an EMSL technique, actually done by Alice. She's sitting in the back here. It's an antibody-driven technique where gold particles are linked to antibodies, which then connect to peptides and the proteins that are representative of that antibody. It can be used to show the surface expression of these proteins that are hypothesized to be on the surface and involved in electron transfer. The presence of all these yellow stains here and the black ones here indicate that the targeted protein, MTRC, which was believed to be the terminal reductase, does exist on the outer membrane of the organism's surface. But to confirm that that protein was in fact active and part of a molecular wire connecting to the interior of the cell, an innovative protoliposome system called this, which is over here, was devised for mechanistic studies on the thermodynamics and kinetics of electron transfer from this molecular wire to mineral materials. This was developed as part of the Grand Challenge, and it's a very innovative model system. The model system includes an enclosed lipid bilayer simulating the outer membrane of the organism. It's filled with a reductant shown over here. This one is methylviologen. It has a reduction potential shown here, but this liposome can be filled with all other kinds of reductants that have redox potentials representative of the metabolic cycle of the organism. The molecular wire or protein complex was synthesized by recombinant techniques using E. coli and implanted in this enclosed lipid bilayer in the same fashion, the same physical orientation that occurs in the organism. These materials were then contacted with mineral materials, goethite and lepidocrosite here, and electron transfer monitored over time in kinetic experiments. And over here on the right is the concentration of the reduced reductant, and its decrease with time indicates that in fact electrons are moving from the reduced reductant through the protein complex that's embedded in the protoliposome out to the mineral material and accomplishing this electron transfer process. And this was done for many different mineral materials, for different reductants spanning the redox potential range of the interior of the cell. And this has turned out to be a functional model for how these proteins, how this process occurs. But as you're probably well aware, that a protoliposome is not really what occurs in the environment. And so this process has been studied in natural materials, in a Hanford sediment, and electron transfer between these organisms and mineral materials generates reactive iron-2 species that turn out to be very reactive with technetium-7, which is a Hanford contaminant, but it's also a very effective redox probe. If technetium is added to a sediment that has had in situ biologic reduction, technetium is reduced over a period, in this case, of 120 days. An EMSL technique called Mossbauer Spectroscopy, as performed by Ravi Kukadapu, who's in the audience here, is a very sensitive spectroscopy to investigate the chemical state of iron minerals in a heterogeneous material. And his measurements were able to show that accompanying the reduction of technetium, as shown here, there was a change in the Mossbauer spectra that was directly a result of the consumption of the reactive phases in the sediment and an increase in the reaction products. And these kind of measurements really cannot be done anywhere else and provided insights on the species that were responsible for this reaction in a very complicated, heterogeneous natural material. So our fundamental model studies with protoliposomes, extracted organisms, provided key insights to understanding what was really happening in that natural material. I'd like to shift a little bit to my second example, and that is related to the Hanford site. As many of you probably remember, the pioneers perhaps, that one of the justifications for the EMSL facility was the Hanford site. and providing information, understanding, and perhaps models and parameters that would be useful to the Hanford site. 20 years ago, Hanford was under significant scrutiny, especially for contamination events in the tank farms and also in the river corridor. And we were able, in working with the site, to establish a program when Ernie Moniz was deputy secretary that was called the Vito Zone Groundwater Integration Project. That project was a partnership between the EMSP and ERSD program and the Hanford site per se, where dollars were shared. And that program took samples from very high visibility Hanford sites and investigated them in the laboratory with the idea of understanding really what was going on and providing insights to drive decisions about what to do about those sites. And over that time period, this program, through EMSOL, looked at 6 different highly visible Hanford sites. One of those is shown here. And it's an infamous event that occurred in the BX-102 tank farm, where over 10,000 kilograms of uranium was released to the Vito zone in a single event. That's a lot of uranium. And with the drinking water standard in the part per billion range, or micrograms per liter, one could envision that this amount of uranium, if it went into groundwater, could contaminate over 10 to the 12th liters of water. Okay, so it's not an insignificant amount of uranium. This figure shows what was known or thought about the disposition of that uranium at the time we did this, or at the time that this work was done in EMSL. You can see the question marks on here. And the fact that there are question marks on this document indicate that there are a lot of question marks. You can see them all over the place. This released materials was and continues to generate a sizable uranium plume in the 200 area. So it's a problem of significant visibility, and also it's one that is currently being addressed, but requires some science to understand really what to do about it. So the team of collaborators, and again, this involved, I think, close to 15 different PIs from the EMSP program and the ERSD program. And we were able to work with the Hanford site and retrieve basically a core that was taken from underneath that leak tank. And by the way, these are the single shell tanks. One of those is BX-102 in construction. You can see that these are pretty sizable features, pretty sizable volumes. And so along with the Hanford site, we basically negotiated a series of science and technology issues that would be addressed. That included understanding the valence and chemical state of uranium, its physical distribution and mineral residence, and its future migration potential. At the time that this core was taken, you can see that the center of mass of the uranium was about halfway to groundwater. Groundwater is right here. So an important question was, is this stuff stable where it is? Can it move? And if it moves, what will the concentrations be and what volumes of water might be required to do that? And as part of this whole concept of wraparound science, the objective really was to do science with impact beyond Hanford. That is, to answer the question, provide some insights, help solve the problem, but at the same time investigate scientific issues with significance well beyond the immediate objective. So one of the first things that was done in EMSL was thin sections of grains from those materials were investigated using electron microscopy, and a really curious finding was observed, and that is that much of the uranium was found inside mineral grains in fractures. It had migrated internally. The white stuff is the uranium. It had precipitated in cracks. And beyond that, it had precipitated in discrete domains. These darker domains, and not the lighter domains, and those reflect a different mineralogical environment. And so that was the first observation that was really quite curious. And so going back to my first slide where I talked about physical environments, you can see that in this case, the physical environment is critical. It's not on the surface like you'd think probably normally, but rather it's in these intergrain cracks and other details like that. And so at that time, EMSL had, Don Baer, I believe, had just gotten focused ion beam sectioning. And we were able to use that technique in some of its first applications to extract thin sections, to basically carve them out, to look at the nature of the fractures where the uranium was precipitating, and then to take those thin sections and look at them with TEM to understand the structure, morphology, and domain size of the precipitates, and then to image them to get insights on their chemical composition, which we did. And these all provided information that was important to understand, let's say, the solubility of these uranium precipitates, or in other words, the concentrations that they would release into water under ideal conditions. But these are not ideal conditions because they're in fractures. So we were able to also work with Paul Majors and with the NMR group to try to make direct measurements of diffusion rates in cracks and pores and fissures in these materials. It was an application that had only been done a couple times. There are real challenges in terms of getting lithic grain fragments into the NMR probe and basically preparing them for a self-diffusion measurement with a specific isotope of hydrogen. We were able to do that, excuse me, and we were able to actually make direct measurements of diffusion inside these lithic fragments, finding that there was a high diffusivity domain with a diffusion constant not too much lower than water itself, and a low diffusivity domain with a diffusion rate of close to 2 orders of magnitude below this. And there are a lot of things that one can do with this data in terms of calculating tortuosities for the internal domains. And that was done. And we did find that there were 2 discrete domains and calculated diffusion and tortuosity values. And these were utilized in a microscopic transport model. And I'm not going to go through all of this stuff. But the important thing is that the way this was applied is that experimental measurements were made to look at the release rates of uranium from these materials. You can see that it occurs over time periods of close to 200 days. It's very much a kinetic process. We also looked at the kinetics of dissolution of the mineral phases themselves and found out that the kinetics of the diffusion process itself were the dominant thing controlling kinetics. And so the diffusion values that were measured by NMR were then utilized directly in this transport model to calculate fluxes of uranium from these materials. And this model was eventually linked with a larger scale model and applied to the field to make predictions of uranium fluxes from the deep Vito zone as a function of different water contents that might result from barriers at the surface. And so this information was taken from the beginning to the end, driven by EMSL measurements, providing real and valid insights to what might occur in the field. I'd like to finish up with just this one last figure. As most of you know that are involved in EMSL, that the nature of science being done in EMSL has changed dramatically over the last 20 years. That, as I mentioned in the beginning, the focus was very strongly on legacy contaminants. After 20 years of work, I'd say that that problem has not necessarily been completely resolved, but significant progress has been made. Many of the big questions have been answered, or it's certainly that we could answer with Hanford. And other problems are now taking, I'd say, precedence. One of those is the behavior of carbon and nitrogen in the subsurface. I think Allison mentioned this in her talk. You did, didn't you? Yeah. And over the last 5 years, Nancy Hess and her colleagues here have really been pushing hard on the ability to characterize organic carbon in detail using EMSL techniques. And by the way, this has been something that has been historically very, very difficult. Still remains a real problem. But they have made tremendous, I think, advances in the ability to do this, both through FTICR MS measurements but also through data manipulation and equating the structures of these compounds with their thermodynamic state. And so understanding structure and thermodynamic state allows us then to predict the rates and driving forces for reactions such as microbial respiration of carbon. And so this technique has been applied to a field site near our lab here, where along the Columbia River, where groundwater and surface waters interact. It's a very complicated system. This shows the river stage of the Columbia River during the course of the spring. The river goes up very high, then it goes down. But the point being that at certain times of the year, river water infiltrates into the groundwater very far. That water has a different chemical composition. It has a different dissolved organic carbon signature. And FTCIR tells us that the thermodynamic signature of these different carbon sources is different. And it gives us a conceptual model to understand why respiration is higher under certain circumstances, lower under other circumstances. And it also provides us with an understanding of why the presence of nitrogen, which is much higher in groundwater, greatly stimulates respiration in the zone of interaction. So this is a new area. It's one, at least for many of the investigators here. I know Nancy and her team has, I think, many users from outside using these techniques for their own system to understand these kind of controls. And it certainly represents an area of growth for EMSOL and for application to the subsurface environment. So I'd like to conclude with this figure. Maybe identify just a few new science challenges and opportunities. Obviously, it's from my perspective. But as you look at a place like EMSL, there are so many capabilities that there are a lot of opportunities. And environmental science continues to evolve. And environmental science will continue to find the capabilities of EMSL at the forefront. And the ability to look at these things in a multimodal character over different scales and different time domains to be a very unique capability, not yet documented elsewhere yet. So some things that just come to mind. First of all, this organic matter characterization thing and really understanding this material in far more detail. It's been a bugaboo in the environmental sciences for over 100 years. Lastly, I'd like to just mention this whole concept of porous media structure. In my first slide, I showed how the subsurface was structured at different scales. And we still don't really understand the interaction of physical structure with microbiologic activity and with chemical reactions. EMSL does have some techniques in terms of Fib tomography that are perfectly suited for this. It turns out that the reconstruction methods are very challenging. And so improvements in reconstruction methods and also devising sensitivities to organic carbon and to microorganism presence, I think would be a really important and great area of focus. EMSL has also in their computing facility has a multi-scale computing activity. Tim's over here. Part of their activities have been verifying and documenting pore scale models. Having this kind of information, and by the way, purple here is organic matter. I forget what the other colors are here, but the important thing is that in natural materials, these different reactants are distributed in a very complicated way. The pore structure is aligned sometimes with them, sometimes against them. Sometimes waters come their way, sometimes they don't. You don't really know. And so having this kind of information can really provide mechanistic insights on the behavior of these systems. And this is just a listing of some of the things that I think are accessible. Certainly they're beginning to be tenable given EMSL's advance in this and new capabilities. And that includes pore networks and connectivity. It's actually a very complicated issue to come up with that information. Understanding where reactive things are and their properties that are actually exposed. Understanding where carbon species are, bound carbon species are, and perhaps their functional groups that are exposed to water or consumed by interaction with the surface. And lastly, the distribution and composition of microbial communities and factors controlling their connectivity. Do the communities connect? How do they connect? What are the mechanisms of connection? Is it transport or are they biologic? And so these are some questions that I think are on the threshold for the facility and would be really interesting to look at. So I can only say from a personal note at this time that over the last 25 years of my career in using EMSL, that it has been probably a career-determining activity for me in terms of access to certain things. And over that same period, I've been advisor to the Stanford synchrotron light source, where they do a lot of synchrotron measurements, XFs. And in reviewing all of those proposals, I'm intimately familiar with what they've accomplished there, what they've been able to accomplish. And the distinguishing factor of EMSL is its versatility to look at physical domains, chemical domains, microbiologic domains over a really large range of scales. You really can't get it anywhere else. So, rah, rah, EMSL. Let's celebrate and let's hope for another great 20 years.
You spent quite a bit of time, first part of your presentation, talking about how electrons might be transferred from microbes to surfaces. And then in the second section of your presentation, you talked quite a bit about what's happening in microfractures at Hanford. And I realize that the second area that you looked at is largely aerobic, but is there any evidence that iron reactions play a role in retention of radionuclides such as technetium in these microfractures?
Oh, the microfractures. There is evidence. There is evidence in the river corridor in what we would call microniches in hyporheic zone and nearshore sediments that technetium reduction is active. And the reason for that is the incorporation of organic matter into fines that represent the infilling of coarse riverbed materials.
Nice.
In terms of the fractures like that were holding the uranium, we did get access to a deep technetium, a deep core that traversed a technetium plume in the 200 area into the upper Ringgold Formation, which we expected and hypothesized would reduce a portion of the technetium, because that's an area where technetium is missing. But we could not document any reductive potential of those sediments from that core. That's the only time we were able to get, I'd say, valid samples. And as you well know, these samples are very expensive to retrieve, and we're at not the mercy, but we have to get them from the Hanford site, and those activities are very costly to perform.
John, I think you have been one of the most successful scientists in my experience at Hanford in having access to those samples. And I know that it's involved a great deal of personal interaction with people on the ground involved in these sites. Reclamation. Thank you.
Well, thank you. I appreciate that. Yeah.